Methotrexate is a chemotherapy drug that is commonly used to treat cancer. It works by inhibiting the activity of an enzyme called dihydrofolate reductase and is also involved in the synthesis of DNA and RNA. Specifically, methotrexate interferes with the conversion of dihydrofolate to tetrahydrofolate, which is required for the synthesis of nucleotides. In terms of the cell cycle, methotrexate primarily affects the S phase, which is the phase of the cell cycle where DNA replication occurs. By inhibiting the synthesis of nucleotides, methotrexate prevents cells from replicating their DNA and progressing through the S phase. This ultimately leads to cell death and a reduction in the size of the tumor. The article that I chose for methotrexate was a systematic review comparing experimental design of animal and human methotrexate efficacy studies for rheumatoid arthritis, which are lessons for the translational value of animal studies. The key question asked is how reliable are animal studies as predictors of the efficacy of methotrexate in humans with rheumatoid arthritis? The experimental design, which is shown on the right, shows a word cloud of the words that were used to describe the experimental designs in human studies. When explaining the data in figure, it shows the frequency of reporting of randomization, blinding, and power calculations in animal and human studies of methotrexate efficacy for rheumatoid arthritis. When interpreting the data, the figures show that randomization was reported in 38% of animal studies and 78% in human studies in figure A. Blinding was reported in 13% of animal studies and 68% of human studies in figure B. And lastly, power calculations were reported in 3% in animal studies and 26% in human studies in figure C.
Paclitaxel is an anti-mitotic chemotherapeutic. This drug functions to suppress the polymerization dynamics of microtubules, which ultimately causes mitotic arrest. Paclitaxel's mechanism of action involves binding to the microtubules in place of tubulin dimers and promoting the assembly of alpha and beta tubulin subunits in order to stabilize the microtubules. This stabilization allows for lengthening of the tubulin polymer, which interferes with the microtubule dynamics. This ultimately leads to mitotic arrest upon insufficient requirements to trespass the mitotic checkpoint, leaving the cell stuck in the G2 or M phase.
This figure is from the scientific paper Testing Macrophages for Enhancing CD47 Blockade, Elicited Lymphoma Clearance, and Overcoming Tumor-Inducing Immunosuppression. Here for this figure, the experimental design was to use in vivo models to determine combination of ACD47 and paclitaxel efficacy by transplanting Raji, Dowdy, or SUDHL2 cells into experimental mice in order to establish the non-Hodgkin's lymphoma, and subsequently treating them with CD47 and or paclitaxel. The key question for this figure is, does paclitaxel resynthesize the response of ACD47 in mice with NHL? And in this figure, in figure 3A, these are the locations where the transplanted cells were placed in the mice. And in figures 3, B, C, and F, these figures, you can see that the treatment with only ACD47 or just paclitaxel, the tumor size was reduced only slightly. However, whenever they were combined, the tumor size significantly reduced.
Herceptin is a monoclonal antibody used in treatment of certain types of breast cancer that overexpress the human epithelial growth factor receptor 2, or HER2. HER2 is a protein that plays a role in the regulation of cell growth and division. Overexpression of HER2 is associated with more aggressive forms of breast cancer. Herceptin acts by specifically targeting the HER2 receptor distributing signals pathway involved in cell growth and survival. By inhibiting these pathways, Herceptin interferes with the progression of cell cycle, particularly the G1 phase, and enhances the immune system ability to eliminate cancer cells through ADCC. This targeted approach makes Herceptin a valuable therapy for HER2 positive breast cancer. The mechanism of action of Herceptin involves several steps, and its primary target is the HER2 receptor, as we discussed above. And here's how the Herceptin works in relation to the cell cycle. Binding to the HER2. So HER2 is a tyrosine kinase receptor that, when activated, signals the cell to grow and divide. And Herceptin specifically binds to the extracellular domain of the HER2 receptors and inhibition of signal pathways. By binding to the HER2, Herceptin inhibits the signal pathways that promote cell growth and survival, and this intervenes with the signaling that helps to slow down or stop uncontrolled growth of cancer cells. This is figure 1 from the research paper Long-Term Herceptin Treatment in the continuation study of patients with HER2 positive breast cancer or HER2 positive gastric cancer. And this shows the worst left ventricular ejection factor or LVEF for each year of treatment. The primary objective is to provide Herceptin therapy to patients with HER2 overexpressing locally. And the ROPE study was a single-arm, multicentral international continuation trial of the Herceptin in patients who had previously completed the global Roche-sponsored trial with Herceptin therapy and had stable diseases and were receiving Herceptin at the end of the lead-in trial. Patients with chronic heart failure during the lead-in trial could be included in following a risk-benefit analysis. In this figure, N is the number of patients with LVEF assessment on study with the year on Herceptin. In case of multiple LVEF assessment per patient within the same year, the lowest value is reported.
So my question is for this slide is describe the mechanism of action of avatism. So for cancer treatment relating it to cell cycle and specific step of the cell-to-cell cycle. Avastin, also known as the bevacizumab, is a type of targeted cancer drug treatment. Depending on the cancer type, one might need combination of this drug with another drug. The way it works is by targeting the cancer cell protein called vascular endothelial growth factor, also known as VEGF, which is a protein that helps cancer to grow blood vessels so they get food and oxygen from the blood. Avastin is arrest proliferation of VEGF, enrich choroidal endothelial cells by stabilizing the cell cycle of G0 and G1 phase and inhibits the G2 M phase in a dose-dependent fashion. So the name of the article that I use is the utilization of bevacizumab is in patients with advanced ovarian cancer, a systematic review of the mechanism of and effects. So the key question for this article is to summarize the molecular mechanism of action, safety, and effects of bevacizumab for use in advanced epithelial ovarian cancer. The experimental design is to see and compare the binding of VEGFR2 with avatism and compare the complex activity signaling cascade mediated by MAP kinase and PI3K